done, and then we went on to do some genotyping with exo-length, and also we have some children have 3-year follow-up. So all in all, we have done this genotype studies. So I published all this, but then we went on to do genomics, and then we conducted GWAS of these children. Some have received atropine, and some have responded. So we have done this, and then the data have been out, and now we are working on replication. So it's not published yet. So we haven't published this data, and we have data in the longitudinal studies, 3-year data, 1,000 study subjects. So we have this needs, and now we are doing the validation in another cohort. So that's what we've been doing. Also ocular trace, corneal astigmatism, and the corneal curvature, and all this detailed endophenotype. We also did meta-analysis GWAS for that, and the myopia and trait, and also the genomics for atropine treatment, the low dosage atropine, the responses. The study was on 262 children. And then we have the treatment response, and we can combine meta-analysis. So we have all these needs now, and then we are organizing a study for the atropine treatment for a validation, right? And we have the treatment responses. So we have conducted a randomized control trial published 2 years ago, and now we are replicating this genetic GWAS list in the LAM2. This another stage of our another phase of our LAM studies, right? And the number of study subjects went up to 400. So we're doing that. So our conclusion is we have so far have accumulated some SNPs with significantly associated children baseline equivalent, and then we have association baseline AL, exo-length, and corneal curvature, and there are suggested associations with some multiple SNPs, multiple genes, but they are all under validation now. So hopefully, give us another several months, we'll complete that, and we should be very glad to publish some of our findings. So I acknowledge the team that in Hong Kong, in the department that work on this, and then we do need not just validation, go on to do deeper. So I've called for collaboration, all right? And welcome to Hong Kong in the next APAO in Hong Kong, and this is Hong Kong at night. So thank you very much. Thank you.
Thank you, Dr. Kelvin. You have some, yeah.
All right, I have one question, and that is the use of cell phones, tablets, children now reading, they even read books, believe it or not, and I see it more in the Asian children that have a higher incidence of myopia. So I'm wondering if the usage of their eyes at near is also a contributing factor, and this also may spread to other areas of the world. There's sponsorship in India at the Prasad, I think at Rao's Hospital that sponsors studies in myopia.
Thanks very much. Very important question. And this is what we call near work, right? Whether it's tablets or screen and books, right? So we have data on that now because of these questionnaires. And actually, in one study, we also have sensors, right, that can measure the reading distance. So we have information on that, but it is quite very clear from what have been published and some of our recent data. Yes, definitely near work is associated with childhood myopia, right? That's definitely. The other, of course, is outdoor time as well. Yeah, you're right. Thank you.
Any other comments, Dr. Kumar? Dr. Vinay, yeah.
If there are patients who have a genetic predisposition to myopia, would atropine treatment help in such patients?
It should. It should. But of course, we are going to have all the information, right? So from our atropine study cohort, of course, we have the phenotype and then the responses of treatment, and of course, then we will genotype all the known related genes or SNPs. Yeah. And regardless, I think regardless of the genotyping, they should have response. But then the response may have, association with some genotype. So again, we will going to find it out. Right? later. Thanks very much.
So Kelvin, I think when they say that you should play 1 to 2 hours in the sunlight
Yeah.
during the sunset. Maybe Kumar, me and Jenny played a lot, so we don't wear glasses.
Right.
Okay. So Kumar and me are classmates and we don't wear glasses. All our classmates wonder how come.
Well, actually, I wear glasses just to see.
All right. Okay.
Okay.
Thanks very much.
Thank you, Kelvin, for coming all the way. Thank you. So next we have a John Prakash recorded talk. No. Then Dr. Musa Khan. No. Then. So Dr. Schultz next, and Dr. Jenny is also then. Okay. So next is we have Dr. Gerald Schultz. We call him Jerry. He is coming to All India Ophthalmic from 1983, I think. And I remember at 83, I came for a meeting when I was a postgraduate, and almost 40 years he is attending almost every year. So Jerry is a All India Ophthalmic Society honorary member.
Yeah, there's a personal interest that I have in ophthalmic genetics because members of my family, we have congenital cataracts inherited in my family. Actually, it's my wife's family. And also I have patient lines with various genetic conditions like microcornea. Other conditions too, and glaucoma we study. So this has personal meaning. And yet on the airplane coming here from the United States, the woman sitting right next to me, her child has retinitis pigmentosa. How do I know this? Because she saw me with my laptop going over my talk, and that started a whole conversation. And I happened to have the literature on that in my computer, so she was studying the umpteenth amount of literature on her child's condition while we were flying over here. So it's very personal. So let's see. We are going to take a look at where we are today in genetic treatments. Of course, I have no financial interest in any of the manufacture of genes. Here's a list of the various genes that can cause ophthalmic disorders. They're huge. And it turns out that about 90% of conditions that affect the body will have some representation in the eyes. Now, we've got to take a look. There's also an elephant in the room. It's hard to ignore that. Now, let's go into some definition. Genetics, of course, is the study of specific genes and their DNA and RNA for inheritance, et cetera, for specific therapy for them. But genomics is the study of all the body's genes, the whole genome sequencing. I just want to mention that because ophthalmology, as I showed you, is part of genomics. Gene sequences, for those realize it's only a tiny portion in a chromosome, and for RPE65 it happens to be chromosome 1, and then spot is number 1. There are a lot of new technologies for determining where the genes are precisely, and that's very important. Now, this is at Cornell. They put in this system, so this is for the study of genomics. And where I am in San Diego, we have the Singlet Genomic Sequencer that very precisely, very quickly could analyze and spot the DNA and the genes in the whole body. So that's not an issue right now because we have that. Now the issue is how we're going to treat the problems and what vectors we have that can go straight to the offending mutation and correct it. The home testing kits, here's a list of them. Oh, by the way, 23andMe is now bankrupt and out of business. Again, I'm showing you all the gene variants that affect the eyes. And with that, let's take a look at the naming of it. The Hugo Gene Nomenclature Committee has set up names for the known human genes. They've named about 19,000 of them. There's still about 25,000. And the reason is that a lot of researchers put their own names on the genes that they're working on, but the Hugo is trying to stabilize it. Now, Craig Venter is in my area. It says, yes, he was in Boston, but his plant is on the campus of University of California, San Diego. And I mention him because he has worked on with the idea that you change the DNA software, change the species. It's very much like the computer binary code. You change some of the coding, and of course you get different codings for whatever you're working on. Now here's some of the options for treatment. We have the viral vectors and its associates, adeno-associated virus, and the non-viral strategies. Ribosomes, antisense, suicide gene therapy, and CRISPR, which I'll talk about a little bit more. So the adenoviruses are non-enveloped, double-stranded DNA viruses that can infect a broad range of human and non-human cell types, and they could be used for genetic treatment. And then we have the encapsulated variation, and the most popular one is the adeno-associated virus vectors, because they could actually penetrate the membranes of the nucleus and get to the DNA. There's the non-viral vectors. We've got the growth factor gene therapy to enhance the survival. This is for retinal photoreceptors. And we have the anti-apoptosis gene therapy so that the tissue does not die. It slows down the death of the cells of the proteins for the photoreceptors. Then we have the suicide gene therapy to knock out totally an offending gene or DNA. And then the HSV transducer cells, this is, believe it or not, with the medication, ganciclovir, which I use quite a bit, because this can also affect the genes to keep them from the herpes simplex and the alterations that virus can cause. So here's some gene therapy, the vectors that can be transmitted. And I did mention the AAV, and the most popular one is 2. It's most workable. And that one, it's used for the inherited retinal dystrophies. I did mention the biallelic RPE65 mutations on the short arm of chromosome 1, and it's used for retinitis pigmentosa. It's also affecting LCA and the fundus elegantus. So this is how it works. The gene is placed in the AAV2, and that is then breaks through the membranes